fertilization and embryonic development. In that, first of all, we'll focus on fertilization topic. Just we'll look forward towards the fertilization today. The fertilization. The fertilization refers to the fission of male and female gametes. Like male gamete, we say spermatozoon, and the female gamete is considered as ovum. It generally takes place in the middle segment at ampullary-isthmic junction. Ampulo-isthmic junction. We call it as fallopian tube. That fertilization involves the following events. So before we go the fertilization, let us see the situation of our said ovum in the fallopian tube. The secondary oocyte is there, which is at meiosis 2 and metaphase 2 stage. A periviterline space you can see here, and then after zona pellucida, then corona radiata, that cells of the corona radiata we can see here. So this is the condition, this is the situation here in the fallopian tube. Secondary oocyte is yet to go for meiosis, to completion of meiosis 2, but at present it is at meiosis 2 and metaphase 2 condition. In that situation it is present. Okay, so then before fertilization, the ovum and the sperms reach the ampulla for fertilization. Here, how? That means we need to discuss about the copulation. As we said about the columns of corpora cavernosa and corpus spongiosum in the transverse section of the penis, whereas during coitus, that into these columns of corpora cavernosa, when the blood is filled up, the penis becomes erectile and going for the ejaculation into the vagina of the female, ejaculation process. The insemination we used to say here, the release of sperms into the female vagina, insemination process we can say. So during insemination, the sperms will enter into the vagina and travel through the cervical canal. We'll watch the video, how do they travel towards the cervical canal and how do they reach to the fallopian tube after all the completion of this. So then fertilization can only occur if the ovum and the sperms are transported simultaneously to the ampullary isthmic junction. That means we know that from the ovary after the ovulation, the secondary oocyte along with the zona pellucida and corona radiata, it will travel here. It will travel into the ampullary isthmic junction. At the same time, sperm should come to that particular fallopian tube for fertilization. Okay, so this is one of the reasons why all that copulation leads to the fertilization and pregnancy. We are not going to say that all copulations cannot lead to the fertilization. Why? That because when a copulation occurs, when the insemination occurs, when the sperms enter into the fallopian tube at the same time, that secondary oocyte should also send that ovum, so-called ovum, we call it as ovum actually, but that is secondary oocyte. That should be there in the fallopian tube at the time. That at what time it will be there, that we discussed in the menstrual cycle. We just said there 14th day that ovulation occurs and that secondary oocyte can be expected there. It may be that exact days we can't calculate in a proper way, just learned as a method. But between the 10th day to the 18th day of menstrual cycle, in the menstrual cycle, in between them, there are more possibilities of the presence of secondary oocyte or ovum is in the fallopian tube. At the time when copulation occurs, there are all the possibilities of the fertilization. Possibilities of fertilization is going to be more at that particular time. Then at the time of ovulation, secondary oocyte is directly expelled into the peritoneal cavity. At the time of ovulation, the secondary oocyte directly expelled into the peritoneal cavity, then enter into the fallopian tube. So let us see the mechanism of transport. The fimbriae of the fallopian tube are internally lined by ciliated epithelium. Fallopian tube is lined by ciliated columnar epithelium. We learned in earlier classes. When ovulation occurs, the fimbriae of infundibulum, We are talking about the oviduct. These are the fimbria of the infundibulum. Encircle the surface of ovary and pick up the ovum and then direct it towards the ostium by the continuous beating of cilia. The contractions of the smooth muscle fiber present in the fallopian tube, here we are talking, fallopian tube, also help in the transport of oocyte, and these contractions are increased at the time of ovulation under the influence of a hormone called estrogen. Okay, so that makes the secondary oocyte from the coelomic cavity of the female to the fallopian tube. The released secondary oocyte contain 23 unpaired chromosomes surrounded by inner, we say haploid condition, n equal to 23. So 23 unpaired chromosomes surrounded by the inner membranous layer called zona pellucida, a glycoprotein layer that consists of glycoprotein, and on its outer side surrounded by corona radiata, a secondary oocyte surrounded by zona pellucida, then surrounded by corona radiata. So that cells are arranged in multiple layers like this. These cells are held together by matrix that we are talking about, the surrounded by corona radiata containing granulosa cells. So these granulosa cells are arranged in a multiple layer format, that all the cells, these cells are together. One cell is connected together by an enzyme called, that substance called hyaluronic acid. Okay, so remember this substance. We need hyaluronidase enzyme to break these bonds. Okay, so let me say once again. So, talking about, this is secondary oocyte or so-called ovum. Okay, so then surrounded by zona pellucida. Then the cells of the granulosa, corona radiata cells we are talking here, that one each other bind by hyaluronic acid. The cement which is there in between them is hyaluronic acid. So that means sperm want to go inside, they need to use an enzyme called hyaluronidase. Okay, so that we'll discuss in fertilization. Here, zona pellucida is also known as primary egg membrane, and corona radiata is considered as secondary egg membrane. Right? Zona pellucida, primary egg membrane, corona radiata, secondary egg membrane. Okay, let's just see here. This is the secondary oocyte. Okay, so then that's surrounded by a little yolk substance, perivitelline space we are talking, and that is surrounded by zona pellucida, a layer called zona pellucida you can see, and cells of the corona radiata. So these cells of the corona radiata, we are saying that one each other connected by hyaluronic acid. Okay, so this is the structure of secondary oocyte. If you remember that if we add membrana granulosa and theca interna and theca externa, if we add that, that will become Graafian follicle, isn't it? But what happened? What happened? That from the ovary, the ovulation process released all the secondary oocyte along with the zona pellucida and corona radiata. So who left over in the ovary? In the ovary, that who left over? That theca externa, theca interna, and membrana granulosa. Then slowly it will turn into a yellowish substance. What do you say? Corpus luteum. Okay, so this is the release of secondary oocyte. That what we said now. After the release, the fimbriated funnel catch them, and the movement of the ciliated columnar epithelium of the fallopian tube, the movements of cilia will bring them towards the destination. Who is the destination? Ampullo-ischemic junction is the destination. Okay, so the fate of secondary oocyte, how many days it will remain like that in the fallopian tube, that for sperm, for fertilization? It is held up at ampullo-ischemic junction for 2 to 4 days only. During this period, it is viable, sperm penetrates it, then fertilization takes place, leading on to pregnancy. On the other hand, if fertilization does not occur, the oocyte will dies out and degenerate. As we discussed in the previous classes, that oocyte will die and degenerate, followed by in the ovary also that multiple events occurs. Whatever we said, that corpus luteum, that also going to degenerate in the form of corpus albicans, and uterus endometrium layers are also going to be degenerated over. So that release of uterine endometrium layers we called as menstruation. At what time all this occurs? If the fertilization does not occur, if the oocyte dies and degenerate, corpus luteum will degenerate and in the form of corpus albicans, and endometrium layers of the uterus will degenerate, disintegrates and release out through the vagina. We call this as menstrual cycle, and that is menstruation, release of that particular endometrium uterine layers. Okay, so this is a fate here. Next, the transport of the sperms in the female genital tract after the ejaculation, several million sperm, as we learned, 200 to 300 million sperms for ejaculation get deposited in the vagina. They get deposited in the vagina. So out of these, 100 to 150 sperms manage to reach to an ovum. What happens in between them? That so many the sperms are going to be killed over because of macrophages of the female, because of the cilia may trap them. So that's why all they may not reach toward the ovum. So one is able to penetrate. Among them, one is able to penetrate. And this is because of several factors that affect the transportation and accessibility of the sperm to the ovum. Okay, so then the capacitation, we have to discuss about the sperm. In sperm, the capacitation, the freshly ejaculated sperm, unable or poorly able to fertilize. Why? Because they must first undergo a series of changes known as capacitation. What is capacitation? Capacitation is associated with the removal of adherent plasma proteins and reorganization of the plasma membrane, lipids, and proteins. Okay, so here that simply what do you say here, that each sperm, to prevent the motility of the sperm, over motility of the sperm, that is associated with the adhesive seminal plasma proteins, yellow color I drawn here. Why is that kind of arrangement? That means from the seminiferous tubule to the retitestis to the vasa efferentia to the epididymis to the vas deferens, along the length, when they are transporting, if the sperm start motility, that motility of the sperm happen rapidly from there onwards, whatever the energy they have, the limited energy, isn't it? In the middle piece, we said that they have some energy, mitochondria, an axial filament in tail, that we discussed about the sperm structure earlier. So they need to reserve the energy that to travel into the fallopian tube from the vagina of the female. So that's why. So their movements are limited over. When they enter into the female, now the capacitation, remember capacity increase of capacity, we call capacitation. What happened? Capacitation is associated with removal of adherent plasma proteins and reorganization of the plasma membrane, lipids and proteins. So it also seems to involve the influx of extracellular calcium and increase in the cyclic AMP process and decrease in the intracellular pH. That brings what? That brings the active movement of the sperm. Then capacitation occurs while sperm reside in the female reproductive tract for a period of time, as they normally do during the gamete transport. The length of time required varies with species, but usually requires several hours. The sperm of many mammals, including humans, can also be capacitated by incubation in certain fertilization media. That's what we call the process we say as the in vitro method. So that means when we are doing the fertilization outside even, We need to do this outside even. The sperm of many mammals, including human, can also be capacitated. That means removal of that adherent protein surrounding them, making them activation. Simply we say that making the sperm, increasing the activeness of the sperm in the female tract, we say capacitation, increasing the capacity. Okay, so that you can see in the diagram here. The cholesterol coating as an outer layer you can see on the sperm that should be removed over in the series of events in this diagram. This diagram, you see the cyclical changes here. So that increase of motility that you can see here, increase of the motility, that because of removal of that adherent proteins and fat material surrounding them. The sperms that have undergone capacitation are said to become hyperactivated. And among the other things, the display hyperactivated motility. Most importantly, however, capacitation appears to be destabilize the sperm's membrane to prepare it for the acrosome reaction. You see that also acrosome is in the top of the sperm in the head region, isn't it? So that it is also destabilize the capacitation appears to be destabilize the sperm's membrane to prepare it for the acrosome reaction. So what is acrosome reaction? We'll see. The binding of sperm to the zona pellucida is a receptor-ligand interaction with a high degree of species specificity. Here we get answer that kind of curious questions by some people, like if the sperm of that in general, if you see the mammal sperm is almost all going to be the same, isn't it? Either the sperm of dog or camel or cat, whatever it is, eutherian mammals I'm talking. So if a dog sperm, we want to fertilize with a cat ovum, will it possible? So for all that, human sperm with other animal ovum, do they fertilize in vitro conditions or some other condition? No, not at all possible. Why not at all possible? So you just see this species specificity. Binding of sperm to the zona pellucida is a receptor ligand interaction with a high degree of species specificity. The carbohydrate groups on the zona pellucida, glycoproteins, ZP3, we say, function as sperm receptor. That means secondary oocyte and zona pellucida. We have to talk about the zona pellucida surround these. These zona pellucida is having ZP3, the glycoproteins that we say zona pellucida 3, ZP3 means the function as sperm receptors. Specific sperm receptor, it's just like a lock and key mechanism. That means other sperms even comes, other animal sperms even come, they don't receive. They don't receive means no fertilization. So that's what we can say. Okay. So then next. Wait. Then the acrosome reaction. We have to discuss about the acrosome reaction. Let me erase this first of all. The acrosome reaction provides the sperm. The acrosome reaction provides the sperm with an enzymatic drill to get through the zona pellucida. Zona pellucida. So, okay, first of all, let me erase this. Not listening to us. Okay, it's not listening to us. Okay, we do one thing. Okay. So the acrosome reaction provides the sperm with an enzymatic drill to get through the zona pellucida. The same zona pellucida protein that serves as a sperm receptor also stimulates a series of events that lead to many areas of the fission between the plasma membrane and outer acrosomal membrane. Same zona pellucida protein that serves as a sperm receptor also stimulates a series of events that lead to many areas of fission between the plasma membrane and outer acrosomal membrane. Acrosome belongs to the head region. The membrane fission actually an exocytosis and vesiculation expose the acrosomal content leading to the leakage of acrosomal enzyme from the sperm head that you can see in the diagram, that acrosome reaction. That acrosome we discussed earlier, that is the modified Golgi complex of the sperm. So this is containing lot of enzymes, activation of these enzymes and fission of this particular acrosome with the ovarian, the cell membrane of the zona pellucida. What do you call it as acrosome reaction? That simply we say that as a acrosome reaction. Okay. So then, as the acrosome reaction progresses and the sperm passes through the zona pellucida, more and more of the plasma membrane and acrosomal contents are lost because we utilize. By the time the sperm traverses through the zona pellucida, the entire anterior surface of its head down to the inner acrosomal membrane and is denuded. The sperm that lose their acrosome before encountering the oocyte are unable to bind the zona pellucida and thereby unable to fertilize. The sperm that lose their acrosome before encountering the oocyte are unable to bind the zona pellucida. So that's what we are talking about. So you see here, if the sperm lost its zona, that this acrosome, it can't even penetrate the zona pellucida. So how can I reach to the secondary oocyte? So that this reaction is very important. What we are talking about. Okay, so that means that there should be a membrane and that should break down during attachment with the zona pellucida, and that enzyme should release out and that should make some penetrations, that all that event should happen. Okay, so denudate, that means the down to the inner acrosomal membrane is denudate. This is the denudate condition. Okay, so acrosome is present like this actually. So what happened? That layer is gone. That's what we say denudate. But what we are saying, the sperms that lose their acrosome, the denudate happen before encountering the oocyte, are unable to bind the zona pellucida and thereby unable to fertilize even. Okay, so that series of reactions you can see in the given diagram, that the acrosome is, this is the acrosome here. The acrosome is fused with the zona pellucida. Acrosomal reaction is going to happen, and that makes the penetration of that zona pellucida layer, and then the sperm nucleus is started entering. This violet color is the sperm nucleus. They started entering inside into the egg cytoplasm, into the secondary oocyte. Okay, so that you can see. We'll watch them in video also. So that means here you can understand that total sperm is not going inside. Just the fertilization moment here, this is the remaining part of the sperm. You just see here, just stay there. So that only the nucleus of the sperm is entering into the female oocyte, into the secondary oocyte. Okay, so the constant propulsive force from the sperm's flagellating tail in combination with the acrosomal enzymes allow the sperm to create a tract through the zona pellucida. That's what we are seeing, the tract now in the earlier diagram. So this is a tract. Okay. Now then these two factors, motility and zona digestive enzyme. Motility we said capacitation, and zona digestive enzymes we said acrosome reaction, allow the sperm to traverse through the zona pellucida. Then sperm-oocyte binding. Once a sperm penetrates the zona pellucida, that's over. That once a sperm penetrates the zona pellucida and releases the contents of the male pronucleus here inside into the secondary oocyte, this is their own nuclei. Okay, let me change the color for their nuclei, female secondary oocyte nuclei. Once it was happened, it binds to and fuses with the plasma membrane of the oocyte, and binding occurs at the posterior or post-acrosomal region of the sperm head. That post-acrosomal region of the sperm head that I have to show you. So this is the post-acrosomal region. So that description for the diagram is given, that's all. So that path is formed. That's what they are saying. Okay. So the post-acrosomal region of the sperm head. The sperm glycoprotein called fertilin, which binds to a protein in the oocyte plasma membrane and may also induce the fission. Fission of what? Male pronucleus and female pronucleus. Okay. So egg activation and cortical reaction. This is the third event as we are seeing capacitation. Then Zona reaction. Sorry, that acrosome reaction. Then after Zona reaction, now the cortical reaction we are talking about. That prior to the fertilization, the egg is in quiescent state that we discussed earlier. It is in metaphase 2 in the meiosis 2 and metaphase 2 is there. So, upon a binding of the sperm, the egg rapidly undergoes a number of metabolic and physical changes that collectively are called as egg activation. That collectively called egg activation. The prominent effects include a rise in the intercellular concentration of the calcium and completion of the second meiotic division and the so-called cortical reaction. Okay, so they call cortical reaction. The cortical reaction refers to a massive exocytosis of cortical granule. Notice this. We'll get answer for the various doubts which rise here. The cortical reaction refers to a massive exocytosis of cortical granule seen shortly after the sperm-oocyte fission. When the sperm-oocyte fission occurs, the cortical granules contain a mixture of enzymes, including several proteases which diffuse into the zona pellucida following exocytosis from the egg. What these proteases do, the proteases alter the structure of the zona pellucida, inducing what is known as the zona reaction. Okay, components of the cortical granules may also interact with the oocyte plasma membrane. Okay, so what is Zona reaction? Zona reaction refers to an alteration in the structure of the zona pellucida catalyzed by the proteases from the cortical granules. The critical importance of the Zona reaction is that it represents major black. That means simply we say Zona pellucida after the entry of one sperm inside. When one sperm is entered inside, that means when this one sperm send their contents and the nuclear fission is happening, the Zona reactions facilitate that they become very hard and doesn't allow the other sperms to enter inside. So what do you call that is preventing polyspermy. One is already entered. One fertilization is going on. So that we know that millions of sperms are present. Even they try, they can't enter inside. So that's what we say. Zona reaction, how that it facilitate. What is zona reaction? Becoming zona pellucida. In general words, we can say the thickening of the zona pellucida. For what? To prevent the polyspermy, multiple fertilization prevention. Okay, so zona reaction effect is a result of two measurable changes induces in the zona pellucida. What are they? The zona pellucida hardens. Crudely put, this is analogous to the setting of concrete. Runner-up sperm, that means this is the who came first. Remaining all we say runner-ups. Runner-up sperm that have not finished traversing the zona pellucida by the time hardening occurs are stopped in their tracks. We don't allow the other sperms to come in. That's what we are saying simply. Sperm receptors in the zona pellucida are destroyed. ZP3 receptors we discussed earlier. That only receive the sperm, we said, specific receptors for specific sperm of the specific species. Therefore, any sperm that have not yet bound to the zona pellucida will no longer be able to bind, let alone fertilize the egg. One sperm, one ovum concept we are strengthening. So that's it all about. Then following fission of the fertilizing sperm with the oocyte, the sperm head is incorporated into the egg cytoplasm. The nuclear envelope of the sperm disperses and the chromatin rapidly loosens from its tightly packed state in a process called decondensation. I'll show you in the video that in decondensation, in vertebrates, other sperm components including mitochondria are degraded rather than incorporated in the embryo. So simply they use two colors here. Only one chromosome. Imagine in the female one chromosome, in the male one chromosome is present. Imagine what happens. Fertilization means they both become one pair, one pair. Then if you talk about this is sperm, first chromosome of the sperm, second chromosome of the sperm, third chromosome of the sperm, fourth chromosome of the sperm. Okay, so ovum. Let's talk about the ovum. First chromosome of the ovum, second chromosome of the ovum, third chromosome, fourth chromosome. So, what is fertilization? What is fertilization? The first chromosome fuses with the first chromosome of the sperm fuses with the first chromosome of the ovum. That becomes first pair. It's just like I said in the genetics to you, right chapel and left chapel concept. Okay, so that second chromosome of the sperm with second chromosome of the ovum is going to form. So what do you say? Second pair. So likewise, second pair, third pair, fourth pair. How many pairs we have? 23 pairs of chromosomes we have. So that will go on like that. 23 pairs. So that means 23 chromosomes from the father side, 23 chromosomes from the mother side will form 23 pairs of chromosomes in the zygote. That simply we say fertilization. We call the process as fertilization. Okay, so development will discuss, but the fertilization events, once again, let us try to watch in the video that all these fertilization events in the video. As the discussion we are going on with the fertilization process. So after the copulation, ejaculation, or we use the word called insemination, that when the penis become rigid through the blood flow of the corpora cavernosa, through the corpora cavernosa, when the penis let me stop this. Let me stop this. Before that, I may say that when the blood flow in the corpora cavernosa of the penis become rigid, and we know here in the corpus spongiosum that urethra is present here. That through that the sperms will release nearly 200 to 300 millions of sperms are going to be released into the tract, into the female vaginal. So they need to travel here. So they need to travel along the length. That means vestibule, into the vagina, into the cervical canal. You see here, the millions of sperms have started traveling through that. So that sperms are traveling through the cervical canal. They need to travel. So you see there, the material surrounding them, mucus and the glycoproteins are now released out. We call the process as capacitation. And now they are moving toward the cervix here. So cervix. Cervix usually closed. But when the sperms enter inside, that these particular sperms, that by the opening of the cervix, they start entering inside. Okay, so cervical canal, cervical opening. So that opened, and now the sperms entered through the cervix. See that millions of sperms started entering through the cervix. Okay, going on. So they need to travel through the uterus, isn't it? In the mean process of the travel, that millions of sperms will die in the travel itself because of macrophages of the female, phagocytosis they do, and the pH conditions and various reasons make them to go. Some of the sperms will travel, they go on like that. Contractions of the uterus also helpful for them. to travel. They need to travel towards the fallopian tube. Towards the fallopian tube, they just travel, and there what is required to present? There is the secondary oocyte. Okay, so you see, one of the sperm reached already. One of the sperm reached and they started entering into the secondary oocyte, penetrating the layers of corona radiata and then zona pellucida. Here it is the fallopian tube. We know the fallopian tube is ciliated columnar epithelium. Movements of fallopian tube is also helpful for the movements of secondary oocyte as well as the sperms also. Okay, so let us see here. Ciliated columnar epithelium. Moving the secondary oocyte towards the fallopian tube, and now the sperms. You see here, some of the sperms are trapped in the ciliated columnar epithelium, which they can't move forward. Some are engulfed by macrophages of the female, phagocytized. Because this is the foreign body, isn't it? The sperm is the foreign body to the female. So that's why they are destroyed. Now, then finally, some of them able to reach towards the secondary oocyte. You see the yellow cells, they are granulosa cells of corona radiata. So first of all, we need to release the hyaluronidase enzyme to penetrate into the cells of corona radiata. Because the cells of corona radiata bind with hyaluronic acid. Then entered in the zona pellucida. One of the sperm entered into the zona pellucida. So then you see that zona pellucida, zona reactions are happening as long as the sperm is entering inside. Zona reaction is happened, and all the ZP3 receptors, you can see their disappearance. Slowly ZP3 receptors will disappear. You see that whenever that ZP3 receptor are receiving the sperm, the reactions will go on. That means zona pellucida becomes solid, then doesn't allow the remaining any other sperms to come in. Then the sperm reach to the next level, cross to the zona pellucida, and remaining sperms are trying actually to come in. There is no possibility if they can't enter. Now the zona pellucida became like a concrete wall. Even they try, they can't enter. Okay, we entered inside the male pronucleus. So actually, one more event should happen here, isn't it? The secondary oocyte is at meiosis 2 and metaphase 2. So that should happen. And one more, the chromosomes of the male pronucleus also need to be hardened. 23 chromosomes you can see there. So this is a beautiful event that where the male pronucleus, this is the male pronucleus, whereas the chromatin material, whatever present here, that is condensing. As I said to you, the 23 chromosomes, we need to make them their availability to fuse with the fertilization. In the mean period by the penetration of the sperm, you can see here, meiosis 2, metaphase 2 is going to be continued. Why I am using the word so-called ovum from the beginning? Actually, there is no ovum till the time, isn't it? Secondary oocyte is there at meiosis 2 and metaphase 2. That has to complete anaphase 2 and telophase 2 also. Anaphase 2 and telophase 2 also complete over, and then a ovum and a polar body that should be released over, isn't it? So the polar body should be eliminated out, and this male pronucleus and female pronucleus should fuse each other. So that event should happen. Okay, so let us watch that event. How will it happen? So meiosis 2 is going on. The female pronucleus. So that polar body is eliminated, and now the female pronucleus is ready. So what left over? What left over? The fusion. The fusion of male pronucleus of the sperm. And one more thing you need to notice. So that sperm entered, only the nucleus of the sperm entered, as I said to you earlier. Only the nucleus of the sperm entered and fused with the nucleus of the female. That's what I am saying. I am using different colors here. So these are the female chromosomes of the female pronuclei. How many of these kind are present? 23 of these kind and 23 of male. So the fission, what is fission? So this first chromosome of the ovum means mother, the first chromosome of the father. So that form first pair. Okay, so that has a great importance here. You need to understand, if in the mother the last chromosome is with X and in the father the last chromosome is Y, what happens? That the sex will be determined there itself. That what do you say? That X and Y. So that sex will be determined here. So that's why whenever we ask a question, when does the sex is determined? The sex is determined during the fertilization process itself. Now it was declared as male. Imagine the sperm is with X chromosome. The sperm with X chromosome, ovum is also with X chromosome. So the sex will be determined here itself. That means sex will be determined with the 23rd pair, which we called it as allosome. 23rd pair. Okay. So fission is going on. Fission of chromosomes, male and female, 23 pairs, which it has a crucial role to say in genetics. The 23 pairs of chromosomes, the fertilization process is completed. Now, along with the zona pellucida, you see still some sperms are trying to enter, but no polyspermy. Along with the zona pellucida, that corona radiata that is moving on, that corona radiata will be disappeared after fertilization. Then zona pellucida will be remain till the process of fertilization occurs. That will remain here. Okay, so that's it about the fertilization events that happen. Fertilization events. I hope you may enjoy with the original voice also. Let's watch once again.
Fertilization is the epic story of a single sperm facing incredible odds to unite with an egg and form a new human life. It is the story of all of us. During sexual intercourse, about 300 million sperm enter the vagina. Soon afterward, millions of them will either flow out of the vagina or die in its acidic environment. However, many survive because of the protective element provided in the fluid surrounding them. Next, the sperm must pass through the cervix, an opening into the uterus. Usually, it remains tightly closed, but here the cervix is open for a few days while the woman ovulates. The sperm swim through the cervical mucus, which is thinned to a more watery consistency for easier passage. Once inside the cervix, the sperm continues swimming toward the uterus, though millions will die trying to make it through the mucus. Some sperm remain behind, caught in the folds of the cervix, but they may later continue the journey as a backup to the first group. Inside the uterus, muscular uterine contractions assist the sperm on their journey toward the egg. However, resident cells from the woman's immune system, mistaking the sperm for foreign invaders, destroy thousands more. Next, half the sperm head for the empty fallopian tube, while the other half swim toward the tube containing the unfertilized egg. Now, only a few thousand remain. Inside the fallopian tube, tiny cilia push the egg toward the uterus. To continue, the sperm must surge against this motion to reach the egg. Some sperm get trapped in the cilia and die. During this part of the journey, chemicals in the reproductive tract cause the membranes covering the heads of the sperm to change. As a result, the sperm become hyperactive, swimming harder and faster toward their destination. At long last, the sperm reach the egg. Only a few dozen of the original 300 million sperm remain. The egg is covered with a layer of cells called the corona radiata. The sperm must push through this layer to reach the outer layer of the egg, the zona pellucida. When sperm reach the zona pellucida, they attach to specialized sperm receptors on the surface, which triggers their acrosomes to release digestive enzymes, enabling the sperm to burrow into the layer. Inside the zona pellucida is a narrow, fluid-filled space just outside the egg cell membrane. The first sperm to make contact will fertilize the egg. After a perilous journey and against incredible odds, a single sperm attaches to the egg cell membrane. Within a few minutes, their outer membranes fuse and the egg pulls the sperm inside. This event causes changes in the egg membrane that prevent other sperm from attaching to it. Next, the egg releases chemicals that push other sperm away from the egg and create an impenetrable fertilization membrane. As the reaction spreads outward, the zona pellucida hardens, trapping any sperm unlucky enough to be caught inside. Outside the egg, sperm are no longer able to attach to the zona pellucida. Meanwhile, inside the egg, the tightly packed male genetic material spreads out. A new membrane forms around the genetic material, creating the male pronucleus. Inside, the genetic material reforms into 23 chromosomes. The female genetic material, awakened by the fusion of the sperm with the egg, finishes dividing, resulting in the female pronucleus, which also contains 23 chromosomes. As the male and female pronuclei form, spiderweb-like threads, called microtubules, pull them toward each other. The two sets of chromosomes join together, completing the process of fertilization. At this moment, a unique genetic code arises, instantly determining gender, hair color, eye color, and hundreds of other characteristics. This new single cell, the zygote, is the beginning of a new human being. And now the cilia in the fallopian tube gently sweep the zygote toward the uterus, where he or she will implant in the rich uterine lining, growing and maturing for the next 9 months until ready for birth.
So that's it about the events. I hope you better understand when we see this area once again. So I'm for yourself. I'm going for this back again. So this session is just to understand the fertilization process. So let's come back. So let me read this in the faster way as we discuss now that we have seen the video. So that make us to be very clear. So now let me read this. Fertilization. It refers to the fusion of male and female gametes, that is, spermatozoon and ovum. It generally takes place in the middle segment at ampullary isthmus junction of the fallopian tube. It involves the following events. That before fertilization, the ovum and sperm reach the ampulla for fertilization. Fertilization can only occur if the ovum and sperms are transported simultaneously to the ampullary isthmic junction. This is one of the reasons why not all copulation lead to fertilization and pregnancy. And transport of ovum at the time of ovulation, the secondary oocyte is directly expelled into the peritoneal cavity and then enters into the fallopian tube. The mechanism of transport, that the fimbriae of the fallopian tube are internally lined by ciliated epithelium. When ovulation occurs, the fimbriae of infundibulum encircle the surface of ovary and pick up the ovum and then direct it towards the ostium by continuous beating of cilia. The contraction of smooth muscle fiber present in the wall of fallopian tube also help in transport of oocyte. These contractions are increased at the time of ovulation under the influence of estrogen. The structure of the secondary oocyte. The released secondary oocyte consists of 23 unpaired chromosomes surrounded by the inner membranous layer zona pellucida, consisting of glycoproteins, and its outside surrounded by corona radiate, consisting of granulosa cells arranged in multiple layers. The cells are held together by a matrix composed of hyaluronic acid. That here, zona pellucida is also known as primary egg membrane, and corona radiata is the secondary egg membrane. So then it is held up at ampulo-ischemic junction for 2 to 4 days. During this period, if viable sperm penetrates it, then the fertilization takes place, leading on to an pregnancy. On the other hand, if fertilization does not occur, the oocyte dies out and degenerates. The transport of sperms in the female genital tract after ejaculation, several million sperms, average 200 million sperms per ejaculation, get deposited in the vagina. Out of these, about 100 to 150 millions of sperms, 100 to 150 sperms manage to reach an ovum, and only one is able to penetrate it. This is because of several factors that affect the transportation and accessibility of the sperm to the ovum. Okay, some will be trapped in the cilia and some are going to be engulfed by macrophages. Likewise, freshly ejaculated sperm are unable or poorly able to fertilize. Rather, they must first undergo a series of changes known as collectively as capacitation. The capacitation is associated with the removal of adherent seminal plasma proteins, reorganization of the plasma membrane, lipids, and protein. It also seems to involve an influx of extracellular calcium, an increase in cyclic AMP, and decrease in intracellular pH. So capacitation occurs while sperm reside in the female reproductive tract for a period of time, as they normally do during gamete transport. The length of time required varies with species, but usually requires several hours. The sperm of many mammals, including human beings, can also be capacitated by the incubation in certain fertilization medium, what we call in vitro method. The sperm that have undergone capacitation are said to become hyperactivated, and among the other things display hyperactivated motility. Most importantly, however, capacitation appears to be destabilize the sperm's membrane to prepare it for the acrosome reaction. The binding of sperm to the zona pellucida is a receptor-ligand interaction with high degree of species specificity. The carbohydrate groups on the zona pellucida, the glycoproteins ZP3, function as sperm receptors. The acrosome reaction provides the sperm with an enzymatic drill to get through the zona pellucida. The same zona pellucida protein that serves as a sperm receptor also stimulate a series of events that lead to many areas of the fission between the plasma membrane and outer acrosomal membrane. The membrane fission, actually an exocytosis, and vesiculation expose the acrosomal contents, leading to the leakage of acrosomal enzyme from the sperm's head. As the acrosome reaction progresses and the sperm passes through the zona pellucida, more and more of the plasma membrane and acrosomal contents are lost. By the time the sperm traverses the zona pellucida, the entire anterior surface of its head down to the inner acrosomal membrane is denuded. The sperm that lose their acrosomes before encountering the oocyte are unable to bind the zona pellucida, thereby unable to fertilize. The constant propulsive force from the sperm's flagellating tail in combination with the acrosomal enzyme allow the sperm to create a tract through the zona pellucida. Then these two factors, motility and zona digesting enzymes, allow the sperm to traverse through the zona pellucida. Once the sperm penetrates the zona pellucida, it binds to and fuses with the plasma membrane of the oocyte. The binding occurs at the posterior or post-acrosomal region of the sperm head. The sperm glycoprotein called fertilin, which binds to a protein in the oocyte plasma membrane and may also be induced fusion. So the egg activation and the cortical reaction prior to fertilization, the egg is Piscean state, arrested in metaphase of the second meiotic division. Upon binding of a sperm, the egg rapidly undergoes a number of metabolic and physical changes that collectively are called egg activation. The prominent effects include a rise in the intracellular concentration of calcium and completion of the second meiotic division and the so-called cortical reaction. The cortical reaction refers to a massive exocytosis of a cortical granule seen shortly after sperm-oocyte fission. The cortical granules contain a mixture of enzymes including several proteases which diffuse into the zona pellucida following exocytosis from the egg. These proteases alter the structure of the zona pellucida, inducing what is known as the zona reaction. The components of the cortical granules may also interact with the oocyte plasma membrane. The zona reaction refers to an alteration in the structure of the zona pellucida catalyzed by proteases from cortical granules. The critical importance of the zona reaction is that it represents the major black tooth polyspermy in most mammals. So the zona reaction, this effect is the result of two measurable changes induced in the zona pellucida. The zona pellucida hardens. The first thing, zona pellucida hardens. Crudely put, this is analogous to the setting of concrete. Runner-up sperm that have not finished traversing the zona pellucida by the time the hardening occurs are stopped in their tracks. The second, the sperm receptors in the zona pellucida are destroyed, ZP3 receptor. Therefore, any sperm that have not yet bound to the zona pellucida will no longer be able to bind it, let alone fertilize the egg. So, the following the fusion of the fertilizing sperm with the oocyte, the sperm head is incorporated into the egg cytoplasm. The nuclear envelope of the sperm disperses and the chromatin rapidly loosens from its tightly packed state in a process called decondensation. That chromatin is going to be decondensed in the form of chromosomes. In vertebrates, other sperm components, including mitochondria, are degraded rather than incorporated into the embryo. So that's it. Chromatin from both the sperm and egg are soon encapsulated in a nuclear membrane forming pronuclei. Each pronucleus contains a haploid genome. They migrate together. Their membranes break down and the two genomes condense into chromosomes, thereby reconstituting a diploid zygote. Finally, the fertilization results the formation of syncyrion, or we call it as zygote. The two pronuclei of the male and female are fused each other. We call it as zygote. Okay. So, the fertilization and development in the textbook. The motile sperms. The motile sperm swims rapidly, pass through the cervix, enter the uterus, and finally reaches to the ampullary ischemic junction at the fallopian tube. The ovum released by the ovary is also transported into the fallopian tube, where fertilization, which is also called conception in human being. Fertilization is also called conception in human. Okay, so the oocyte cumulus complex of the female is surrounded by zona pellucida. Oocyte cumulus complex of the female is surrounded by a zona pellucida, corona radiata, and cumulus layer. When a motile sperm reaches the ovum, it makes its way through the corona radiata and zona pellucida. In this process, the enzyme hyaluronidase released by the acrosome of a sperm. It is corona penetrating enzyme. Hyaluronidase is a corona penetrating enzyme that plays an important role in dissolving the hyaluronic acid. Here it is, the cells of the corona radiata, which are connected each other by that hyaluronic acid. So that sperm releasing corona penetrating enzyme breaks that bond between the cells, so it can easily penetrate. So next, another enzyme, acrosin. Another enzyme, acrosin. Let's point out only enzymes. So one is hyaluronidase. corona penetrating enzyme. We use it to say. Okay. So which penetrates the corona radiator, cells of the corona radiator. Next enzyme, acrosin. The acrosin released from the acrosome as an effect of acrosome reaction dissolves, digests the zona pellucida, lysis of the zona pellucida to facilitate the penetration of the sperm. So the several sperm penetrate through the zona pellucida into the periviterline space. Only one sperm enters the ovum. We call it as monospermy. This induces the completion of the meiosis 2 and of the secondary oocyte. The second meiotic division is also unequal and results in the formation of second polar body and an ovum. Now we use that word, ooted or ovum. Okay, so the fusion of gametes is called syngamy. The fusion of gametes, we say syngamy or amphimixis. The nuclear union results in the formation of synkaryon, zygotic nucleus. The corona radiata disappears after fertilization. Entry of sperm causes release of calcium, which blocks the entry of other sperms. The parallel reaction is the Zona reaction. Okay. So next in the box item, it was given that how does the sex will be determined, that we discussed. That X chromosome, that the 23rd chromosome is the X in ovum. 23rd chromosome may be X or Y in case of male. So simply if this X of the uh, ovum with X of the sperm, which leads to XX female, or X of the ovum with Y of the sperm, that leads to XY male, that leads to XY male. So sex is determined during fertilization process only. The 50% of sperm among, if 200 millions of sperms are there, 50 million, that 100 million sperms are X containing X type of chromosome, 100 million sperm, 50% are containing Y type. So that's a speculation that what sperm will reach to the female, and that means X type of sperm or Y type of sperm. Who reaches that possibility is always 50% chance. Okay, so that we discuss more in actually in that genetics. So this is all about the events of fertilization. So it is given very little in the textbook here. So when we go in detail, there you can understand the complete procedure. In that way, we took this particular topic today, fertilization, and then after development process. Development process is a very complicated process here. So that we begin the developmental process after these.